It's great that the Bangalore Life Science Cluster campus organizes a series, and in fact, we become, if you will, a serial series organizer recently in the era of COVID. I must congratulate both the comms office, which, with Mahin, Pavitra, and yourself for taking this initiative, and of course, roping us in to bring you some of the content. And here in this series, the Bliss campus welcomes you to the inaugural lecture of this unique public lecture series titled The Human Body Inside Out. So what is Inside Out? Well, every week, a scientist is going to take you through one part of the human body that they research. From the textbook basics to the two latest research, you will get to know a whole new level about yourself. The series itself is hosted by my colleague, Dr. Raj Ladha, who happens to be the head of academics at the NCBS and the Bliss campus today. In fact, he himself studies some part of the reason why you're able to hear all of us today, which is the inside of the ear. He studies the inner ear, and perhaps in one of the series lectures, he's going to reveal exactly what he does. But the Inside Out series is essentially curated for high school students and undergraduates, but all of you are welcome. Invite your friends, family to register and attend the talk so you get to know more about yourself. Our talks will happen every Friday at 5 p.m. When next week, we have Dr. Bhavna Muralidharan from one of our campus institutes, the Institute for Stem Cell and Regenerative Medicine, who's going to speak to you about the brain. In the following weeks, we will have colleagues of ours from across the country, from IIT Kanpur, Dr. Amitabh Bandyopadhyay will talk to you about bones. Then back to our Bangalore campus, we'll have Dr. Dandapani from InStem to speak to you about matters of the heart. So there is a whole spectrum, and in fact, that's just 3 of the lectures we are advertising for the next 3 weeks. But this week, for the inaugural lecture, we have a very special speaker. We have Dr. B.S. Ramakrishna. He currently is director of the Institute of Gastroenterology and Enterology, Hepatobiliary Science and Transplantation at the SIMS hospitals in Chennai. Formerly, he was professor and head of gastroenterology department, the gastroenterology department at the Christian Medical College, that is CMC at Vellore. And he continues to be an active practicing clinician and an endoscopist who literally looks inside you, besides actively conducting research targeting intestinal disease and its relationship to the microbial residence within the gut. Dr. Ramakrishnan is extremely highly recognized, and he's well celebrated. He's a fellow of several national academies and is in fact served as the president of the Indian Society of Gastroenterology and is the editor-in-chief of the Indian Journal of Gastroenterology. He has served on committees at the international level, at the World Gastroenterology Enterology Organization, the Asian Pacific Association of Gastroenterology, and also at many of the committees at the Wellcome Trust. He is widely published, has written many book chapters, and even has several patent applications to his credit. We actually couldn't think of a better person to start this series because he will literally get to the heart of the matter, which is your gut. He will begin with the gut, and it is really a great pleasure for me to have Rama not only agreeing to be here, but also kicking this series off on the human body inside out. I have a gut feeling this is going to be a great lecture. Welcome, Rama. I believe Raj will say a couple of words right now, and then we'll have Rama come on, or am I mistaken?
Oh, it's I'll wrap up at the end.
Okay.
Thank you very much, T2.
Thanks, Rama.
Thank you. So, may I start then?
Yes, please.
Yes.
So, if I can just share my screen. Thank you very much, Jitu, for that effusive introduction. Thank you so much. It's a pleasure and a privilege to be here. Teaching has always been teaching and communicating science has always been a shared interest, as Jitu knows. And I hope in this lecture, I was just asked to kick off the series. So I was going to set the whole series of lectures in context by essentially by, sorry, excuse me, I'm just trying to get my mistake here. Essentially to get the whole series in context by talking about how science of the human body has advanced over the centuries. And then at the end, I'm going to tell you a little bit about my own special passion, which is the gastrointestinal tract. So before we start, even when I was a child, even when I was in school, I clearly remember that I was very interested in the workings of the human body. And this was in the mid and late 1960s. And in fact, things like neurochemistry had come in at that time. And I learned a little bit about it by reading books outside of school. And that really fascinated me, that we could understand how the body works. And so as time went on, I knew I wanted to be a doctor. I became a doctor. I became a practicing clinician, but I never abandoned my interest in understanding how the body works. And so my research has been on the intestinal tract. And specifically, I've looked at some processes which are involved in transport, interaction of our body with the bugs inside our gut. Now, coming to this series in particular, what are we talking about when we say anatomy and physiology? We are really talking about the structure and the working of the human body. Anatomy, the term comes from the Greek anatome, which means dissection or cutting up, and physiology essentially means study of the function of the body. Now,
Rama, sorry, we're not seeing your screen.
I'm sorry, okay. Still not seeing the screen?
No, we can't. You'll have to share it.
I did share it, okay.
Yes, it's coming now.
Now it's coming, yeah.
Is that okay?
Yes.
All right,
Yes.
Sorry about that. Yeah. So anatomy, as I said, and physiology, essentially, they come from Greek words. So humans have always wanted to advance. And one of the goals has been, can you recreate man? Can you have artificial intelligence? And this gentleman here is Isaac Asimov, one of the most prolific science fiction writers of the 20th century. And these are two of the books he wrote, that's I, Robot and The Bicentennial Man. And I'll show you a little bit later. Have we even got close to what the human body is? The answer is no, the human body is extremely intricately designed. And we are miles from getting anywhere even close to that. So let's start with how people got interested in anatomy and physiology, and how did people understand the structure and the working of the body. So ancient Egypt, This was 1500 to 1000 BC. Egyptians mummified their dead, and this is the 3,000-year-old mummy. You can see that they understood anatomy because of their practice of mummification. Moving on to ancient Greece. Sorry, there's a problem with the full screen. I'm sorry, can you see my screen?
We can't see the full screen, but we're seeing the slide, so it's not a problem. You can continue.
Perhaps go to slide mode instead of presenter mode, Rama. If you go to slide mode, slideshow mode.
Slideshow mode is what I was on, but let me see.
Yeah.
Is that better or just?
Yeah, no, that's better. If you now in this mode, if you go to slideshow mode, oops, you went back to the same same.
This is coming. Oh, I'm not able to switch this thing off.
Yeah, we are seeing your full screen. I think you should be sharing only the PowerPoint, maybe.
So,
Yeah.
yeah, maybe the second option you should share. Yeah.
This one here.
No, the one which only has the slide.
This is where I'm getting to again.
Oh, okay. It wasn't happening when we did the test at the beginning. So,
And I'm not sure why that is so, but
it's okay. We can continue from here. People are able to see the screen. Yeah.
I'm sorry about that. Okay. So moving on to ancient Greece, the Greeks were among the first to objectively study the human body. That's why most medical terms are based on the Greek language. And as gastroenterologists, we always talk about the legend of Prometheus. So Prometheus, 800 BC, he was the person who stole fire from the gods and gave it to the humans. So what did the gods do? They chained him to a rock as punishment, and they got this eagle to peck his body every day and chew up the liver. So during the daytime, his liver was entirely chewed up by this eagle. And in the night, it would regenerate. So that was his punishment. So what it showed us was that they knew about the liver. They knew that it could regenerate. Hippocrates, who came a little bit later, 400 BC, was the father of Western medicine. And essentially, he believed, actually, he did not know that much about anatomy. And his concepts of physiology were a little bit hazy because he believed there were 4 humors. But he is considered as the father of medicine because there's so many things that he said about medicine. The next person to come along actually in the history of medicine was Galen, who lived around 150 AD. He gathered all the Greek anatomy teachings and wrote a book which influenced the teaching of medicine for nearly 1,500 years. And this is the longest lasting influence. So the whole problem was whatever Galen understood, whether perfectly or imperfectly, that was taught to all medical students. So the ancient Greeks believed there were 4 humors. There's the earth, there was water, there was air, and there was fire. And depending on which was predominant in your body, if everything was there in equal amounts, then you were, you were good. But if you had anything which predominated, then your personality was tinged according to that. So as it turns out, the 4 humors of the ancient Greeks were similar to the doshas of ancient Indian medicine. So instead of air in Ayurveda, there was ether and air. But the rest which was the fire, the water, and the earth, were similar. So the ancient Indians were the other people who understood the anatomy and the physiology of the human body quite a bit. And this is a pic of Sushruta, who was a surgeon of ancient India. We really do, the history books on these is very hazy. One surgeon who was called Sushruta. This collection of surgeons, or maybe it was passed down from generation to generation, and they wrote a book called the Sushruta Samhita, which is a textbook of surgery, and that showed a significant knowledge of human anatomy. Now, they described more than 300 bones, but these could have included a lot of bones which are not currently considered bones. They described a lot of muscles, blood vessels, lungs, stomach, and intestines. And these were surgical instruments on the right-hand panel here, which you see, which were described in the Sushruta Samhita. So for that age, it seems apparently reasonably well advanced understanding of gross anatomy. So when the Roman Empire declined, which happened in the 5th century, and up for the next 10 centuries, there was very little scientific progress. And this is called the Middle Ages. This is because everything had shifted to Europe and to Central Asia. And at the end of the 15th century came, or the beginning of the 15th century, came the Renaissance. Now, one of the very prominent people during the Renaissance was Leonardo da Vinci. One of the most multi-talented people the world has ever known. I mean, his talents were, he did so many things. He's well known having painted the Mona Lisa. Now, here is a book on Leonardo da Vinci, his knowledge of anatomy, and it's written by a friend and a colleague of mine, Michael Farthing. He's a gastroenterologist from the UK. And the book essentially shows how much he understood of anatomy and how clearly he's depicted that. He was not primarily an anatomist. He had so many other things to do. He talked about making airplanes, and he painted as well. The first person who actually collected the knowledge of gross anatomy was Andreas Vesalius, also Italian, or well, not actually Italian, Flemish or Belgian, but he taught in Padua, Italy. And it seems that Padua was the center of learning at that time. And he wrote the first complete textbook of anatomy that is called De Humani Corporis Fabrica, or the fabric of the human body. And these are illustrations from his book showing that he knew a lot about the individual muscles that go up to make the body. And there are others which show his entire knowledge of anatomy. Soon after him came William Harvey, who was British by origin, but he also studied at the University of Padua, where Vesalius taught. And Harvey was actually the first person to describe the circulatory system. He wrote a book called De Motu Cordis, which means the motion of the heart. And he showed that the heart pumped blood to other parts of the body. That is the head, the lungs, the liver, the intestines, and the rest of the body. And so these red vessels here are the arteries which are pumping blood to the organs. And the organs and the blood from those organs comes back in the veins to the heart. And he recognized this. Unfortunately, he did not have the tools at that time to understand how the arteries and the veins were connected, because he did not have microscopes to observe capillaries. So at the end, by this time, we could understand that these yellow things here, which are the nerves, that you could put together a framework with the nerves, the arteries, the veins, and the bones, And so you had an entire framework. On top of this, you could put the muscles, and inside you could put the organs. But so we had a very good understanding of gross anatomy at the end of this era. So the next advance in understanding came when microscopy was introduced. You would not even recognize this first microscope which was used, because this is how they used it. They had to hold up the flat object, look at it, and it was basically a lens looking at something, magnifying it. And one of the persons who was a very influential early microscopist was Antony van Leeuwenhoek. You would have studied all about him in school, but this is when he started, and this was early in the 17th century. Soon after him came Malpighi, who applied the microscope. By that time, the microscope had evolved a bit. You can see that it's like this. He put tissues under the microscope and described anatomy there. So he is called the father of microscopic anatomy. Now, he discovered many structures in the kidney. So many structures in the kidney, like the Malpighian corpuscle, named after Malpighi. He was also the person who discovered capillaries, thus linking blood flow between arteries and veins. So that takes us to the end of the 17th century and the 18th century. And in the 19th century, you see, we had Hooke and Leeuwenhoek were in the 17th century. The cell theory was initiated, where people, Schleiden and Schwann, noted that all living things are basically made of cells. And another very famous anatomist or a pathologist called Virchow, he initiated the principle that all cells come from other cells. They do not crystallize from air or water or whatever else, but they come from other cells. So this then kicked off an era at which people had to understand structure and function at the level of the cell. So you had to understand what are the structures within the cell. How does the cell get energy? What kind of machinery does it use to perform its functions? How is everything within the cell regulated or controlled? And how do cells replicate? And this is really what happened after the 19th century. So what improved our understanding of human anatomy and physiology was improved microscopy, starting from the compound microscope, which is basically two lenses put together, then to achromatic lenses, where multiple lenses were put together to increase the resolution. And then we had the electron microscope, which gave us resolution that until then we didn't have. And now we have super resolution fluorescence microscopy, which helps us to understand or visualize things at very, very high resolution. So in the last century, which is the last 50 years of the 20th century, basically people focused on basic cell function. So heredity was something that was focused on. Although Mendel is associated with genetics, during his lifetime, his work was ignored. Only after he died, his work was rediscovered. Chromosomes were discovered, the term genes were coined. We understood what constituted DNA, and that DNA so understood how cellular metabolism occurred. And we understood that most of the structural machinery in the cell is made of proteins. So much so that this gentleman, Sir Hans Krebs, who discovered the Krebs cycle, was actually a hero during our days, because he discovered something very fundamental to life and to humanity. following the 1950s, the last half of this, also 1950 to 1990, was essentially the era of molecular biology, when the structure of DNA was solved, the genetic code was cracked. So we understood that you could have the codons, which is certain arrangements of nucleic acids. So 3 of those would then code for an amino acid. And then we had enzymes which could cut DNA, enzymes which could convert RNA into DNA, sequencing of DNA, invention of PCR, and so on. And from 1990 to now, you can say is the era of molecular medicine, because gene therapies have come into being, the complete human genome is sequenced, and now we have next generation sequencing and the ability to edit DNA. So the reason I brought all this is to make you understand how our knowledge evolved over the centuries. And now we know that you have atoms and molecules, but the basic unit of a living organism is a cell. Multiple cells of different types are organized into tissues. Multiple tissues that serve a particular function go to make an organ. Multiple organs that subserve, for instance, digestion, go to make an organ system. And then finally, you have the whole organism. So at this point, I am actually going to go into a little of the questions about the digestive tract and illustrate to you how intricate this whole system is and how we have been able to understand it at almost very basic levels. So what this video showed you essentially was that sorry, excuse me, let me just go back here. So you can see a bolus of food going down the esophagus, going down, 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 goes into the stomach. Suddenly there's a lot of acid being secreted in the stomach. And then the stomach churns this whole thing so that the food gets made into a kind of paste. Now this is a so food doesn't go down just on the basis of gravity. It goes down because there is a very intricate, well-coordinated mechanism which allows it to go down. Now we understood this mechanism more than a century ago, basically based on physiology at the gross level. So if this is the food pipe, there's a sphincter here, food enters the stomach, there's a sphincter here. Now both of these, they close up and the stomach churns back and forth so that whatever's in the stomach is created into it makes an emulsion. And even the fact that food travels down is a very active process in which a lot of muscles are involved. And these are involuntary muscles which are under the control here of the brain. This is the hindbrain, the medulla oblongata, and the spinal cord. The vagal nerves come out of here and control the foregut. And essentially, in addition to these nerves, there is something called the intrinsic nervous system of the gut, the enteric nervous system. So if you cut open the gut like this, there are multiple layers. There's the layer here which is involved in absorption, absorptive cells. Then there are muscles. Then there's a layer of nerves. Then there are other muscles. Then there's another layer of nerves. And then there's a sheath around that. It's like a coaxial cable in some ways. And essentially, when you look at it, you'll find that these nerves, which are the greenish structures here, are nerves which are there like a mesh around the entire gut. The reddish structures inside are also different sets of nerves which are there inside the entire gut. So what this shows us is that the innervation of the gut is something which very closely coordinates function within the gut. So when you swallow, at the point where the bolus reaches, the muscle has to contract. Just beyond that, the muscle has to relax, the intestine has to open up. Then that area has to contract. Ahead of it, the intestine has to relax. So there's a propagated contraction which goes down, which is called peristalsis. And what is controlling it is this very elaborate nervous system. And if you actually look at what happens within this nervous system, and this is just a pictorial representation, if you look at this as the mesh of nerves which is there, you will find that there are impulses, electrical impulses. If you can visualize it this way, that there are electrical impulses which keep going down from a pacemaker somewhere in the proximal stomach, all the way down through the small intestine to the large intestine. And these impulses, these electrical currents which you see, even the way they are generated is really fascinating, because we understand it at the cellular level, how these currents are generated through the transport of sodium out of the nerve cell. And this understanding is very important, because once you understand this, people can create molecules which target any of these functions. And these are in fact used in treatment. So the next thing that I'm going to talk about in relation to the gut, and something which is very important, which is something which has gained importance in the last maybe 20 years, and maybe is also very topical in terms of the COVID pandemic, which is now going through, is the intestinal immunity. So the intestine is one of the major immune organs of the body. So if you look at this as the lining cells of the intestine, on top of it, there are bacteria. There's a lot of bacteria in the intestine. And underneath the lining cells, you have a lot of immune cells. So the bacteria here on the surface of the epithelial cells talk to the immune cells which are underlying the epithelial cells. And that changes the way our immunity works. Now, what is true for the intestine is also true for the respiratory tract. So the same thing happens when the coronavirus invades the respiratory tract. So that sends signals to immune cells, which then go and influence immune reactions within the body. So let me just show you a video here, which shows blood flowing in the capillaries. This may not be entirely accurate. These are RBs, red blood corpuscles, and these are the white blood cells. So here's a white blood cell, which is adherent to the endothelial cell lining the capillary. It moves along that. As it moves along, it recognizes proteins on the endothelial cell. They get attached a little bit. This white cell flattens out. There are these proteinaceous attachments which you see here. And this is called rolling adhesion, where it keeps moving along through sequential attachment and comes out through gaps between the endothelial cells into the tissues. So this is how cells go from the capillaries into the intestine. These immune cells go from capillaries to the intestine. These immune cells go from capillaries to the lungs. And here is the kind of cartoon depiction of that. So this is the endothelial cell. This blue is the immune cell. So it gets tethered to the endothelial cell. Then it starts rolling along. There are some proteins which are activated. It sticks firmly, and then it extravasates into that tissue. Now, as I said, this happens in the intestine. This happens in the lungs. So how does this white cell know how to get that it has to get out in the intestine and not in the lung? So if it needs to get out into the intestine, these are the kind of molecules, proteins, which it has to deploy on its surface. If it wants to get out into the lung, it has to deploy a different set of proteins on its surface. And these will attach to endothelial cells proteins in the lungs. These will attach to endothelial cell proteins in the intestine. So it's, in essence, it's very targeted the way it goes. And this is one of the marvels of the intestinal immune system. In fact, also of mucosal immune systems, works for the skin, it works for the lungs, and works for the liver, and so on. So just to sum up this part, the body consists of a number of complex systems, with various organs making up each system and a variety of tissues and cells going to make up the organs. The organization of the cells, the tissues, the organs in the entire system is intricate and really beautifully coordinated. So we talked about recreating man. How possible is it? So the book that Asimov wrote, I, Robot, was made into a movie, and here's a clip from that movie. So that is something that probably you'll hear about next week when the talk is on the brain. So whether we can simulate human emotions, whether we'll ever reach that stage, I mean, that happens with many other bodily processes. So before I end, I really wish to just challenge the students here, listening here, and leave you to think about this. Essentially, human knowledge is advanced by challenging dogma. And this is William Harvey, who discovered the circulatory system. As I said, 1500 years of Greek medicine, Galen's medicine. And then he had the gumption to challenge that and said, no, this is how things work. So he said, I've often wondered at those who fancy that everything has been already completely investigated by some mighty name, that nothing could possibly be added to their knowledge. So challenging dogma and lateral thinking is important. When I was a student, and this was before I joined medical studies, there's something called the central dogma of molecular biology. So what that said is that you have DNA, which is double-stranded nucleic acid, that this could create copies of RNA, which was single-stranded, that is called transcription. And this RNA would then go to the ribosomes, where the genetic code would be translated into proteins. So it was thought to be a one-way flow for a very long time. But the central dogma was challenged by these two people in 1970, David Baltimore and Howard Temin, who showed that RNA could actually recreate DNA. There's something called reverse transcription through which RNA could make DNA. So just to sum up, and I've really come to the end here, the human body is an absolute marvel of engineering with extremely complex and intricate systems at the subcellular, cellular, and the organism levels. And I would just tell you that never be afraid to challenge dogma. Lateral thinking advances science and leads to progress. And this series of lectures, which is brought to you by the Bangalore Life Sciences Cluster, I think should be very illuminating. And I hope that you will totally enjoy this series of lectures. Thank you.
Thank you so much, Dr. Rama, for this. I will just ask you before we go to our Q&A session, which Raj will coordinate, to stop your slideshow and also turn off your video, which I think will improve the quality of the audio that is coming through. So and also your screen share.
It's not stopped yet. I'm sorry. I thought okay.
So yes, Dr. Raj will now be taking questions. So please put your questions in the Q&A box and also in the YouTube chat as well. Thank you.
Okay, so thank you very much, Dr. Ramakrishna. I'm going to read out the questions as they arrived. The first one, I think, is very apt for a gastroenterologist. From Usha Priya S. Thank you, Usha. Could you please brief how non-living substances like atoms could give rise to living cells, as theory of genesis from non-living things are disproved?
Yes. So how do non-living things become living?
Yes.
And that is the real question that we still have to answer at the end. If we could, well, we know that everything that's inside us is non-living. For instance, the water that's within us has been around for millions or billions of years. Whatever atoms we have within us probably have been there for billions or zillions of years. How did it happen? I really don't understand it. Perhaps at some point of time, I think Raj, maybe you or the people at NCBS will need to give us the answer to that. I'm more into here in terms of looking at systems and still trying to understand the wonder of those.
Thank you. The next question was from about slide 31, which is the image of the neurons around the intestine. Azim Khan asked if the green color in slide 31 was due to green fluorescent protein.
So it's not an actual capture. It is something that, and it's not the neurons from the enteric nervous system. I just put it there to represent to people, just so people can kind of get a better understanding, or you could say a little more inspiration. It's in terms of knowing what these, you're talking about the animated version, right? The enteric neurons?
No, I think I think it was a fluorescent image.
The other one, that's yes, indeed, yes. It has the labeling on it, and I would have to look at it to see what it was.
Okay.
It's labeled there.
Yeah.
Yeah.
Yeah. So it was I think it was
It is a green fluorescent protein. Yes.
Yeah. So Anurag Yadav asked whether a brain-dead person could perform peristalsis.
A brain dead? Yes, absolutely yes.
Okay.
So brain, as long as there is no circulatory death, absolutely.
Okay. Himanshu Neev, what is a positive Carnet's sign?
I'm sorry?
Positive Carnet's sign?
I didn't hear that clearly. What sign?
Carnet's. Carnet. Carennett. Carnet's sign. We can move on. Yeah.
Yeah.
So Ravindranath G asked, how important is the gut microbiome in relation to the health of an individual?
Gosh. Yeah, that can be answered in one word, extremely important, or you could go on for a long time about it. But essentially, it's something that has evolved with us. And at the NCBSU, people are studying Drosophila, and the microbiomes in the Drosophila gut have co-evolved with Drosophila, with the fruit fly, and so our microbes have co-evolved with us.
Yeah.
And we got our mitochondria from them.
Yes. Yeah. Next question from Sridhar S. Sir, you told that the food paste will move from the stomach to the intestine due to gravity. Doesn't the acid also go into the intestine due to gravity?
No, I said it does not move due to gravity. I said it's a very active mechanism. So maybe it's so one should not assume that it moves due to gravity.
Kruti Vasisatha, why is the gut called the second brain?
So the enteric nervous system is called the second brain of the gut because there's a whole mass of neurons around the gut. So I think next to the brain, this is where you see so many nerve cells all over the place. And the intestines are extremely long. I mean, it's about, say, altogether maybe 40 feet. And you have all around it these nerves which are twined. So there's a huge mass of nerves. That's why it's called the second brain. And the enteric nervous system is involuntary. It functions by itself. It's not under the control of the major brain.
So this, I think, feeds into a question from the YouTube live stream, which was the difference between ENS and CNS, which I think you just explained. Is that?
Yep. I think so. So that's the difference between the ENS and CNS. The CNS. So here, this is entirely divorced from the brain.
Nilima Prabhu, thanked you for a wonderful overview, ending with the fine signals that control digestion. We are now being told that the Indian population has been largely asymptomatic for COVID-19, I guess, since our spices help to protect us against the virus. So could you throw light on this?
All right. So can we protect ourselves from COVID? I think those answers are simple. I think you see it every day on TV. So social distancing and masks are the best way to protect ourselves. Just being sensible. And it's not social distancing, it's physical distancing is what we are talking about nowadays. Other than that, I'm not sure that chemo prevention is going to work. So what you need to understand is that most people with COVID illness will not have disease. or they will not have severe disease. It's only the 2% that has severe disease. That's because their innate immunity goes awry. And instead of working normally as innate immunity should, it starts shooting off cytokines and they end up with the bad consequences.
So the next question asks if we can replicate the human body on a chip, like tissue on a chip, organ on a chip, in order to understand the entire physiology of the human body and for understanding disease.
I hesitate to say no, because obviously human endeavor has always been to that end. And it's certainly not possible now. It's certainly not possible in the next decade. But it could become possible at some point of time. People have started, and you guys are working on stuff like that.
So another question: how do we know when our stomach is full? Which system detects these signals inside the stomach?
So it's basically nerves in the fundus of the stomach and hormonal signals from the stomach, which go back to the satiety center and tell it that, time to stop eating. That's...
Yeah. So the last question I have on the Q&A, no, there's a few more. Azim Khan asked, is there an existence of evidence for gut microbiome and cancer prevention?
Yes, there is. So at least in terms of colon cancer, we think that there is a lot of evidence, but it's not yet translated into treatments or even into preventive strategies at the moment. There's a little bit of understanding, and I think as we go along, we'll certainly have something. But it's much more than colon cancer. I think it's gut microbiome is being linked to a whole lot of things. The only problem is that sometimes people go overboard and they use fecal microbiome transplants in the expectation that it'll cure everything from alcoholism to cancer.
Yeah. What makes the liver unique in its regenerative capability?
For whatever reason, the liver has a huge regenerative capacity. The one, it may be related to the diversity of metabolic functions which the liver subserves. I mean, it does so many metabolic things which help in maintaining body homeostasis. And maybe that's one reason why it developed. So why it developed is something I think that we'll, I don't have the answer to, but we know that it's there.
Yeah. I think people are actively trying to find out what makes the liver regenerative. Is the blood-brain barrier permeable to gut microflora? Asks Sampada Pente.
Maybe not to the best of our knowledge, but that dogma can certainly be challenged. Maybe not to whole microbes as such, but certainly to microbial products. There are experiments which show that microbial products do accumulate in the brain.
Andranka Marut is asking a specific question, maybe about someone they know who has a gastric polyp inside the small intestine, neither growing nor shrinking. What can you suggest about it?
Right. So there are polyps broadly are neoplastic, which means that they could potentially turn cancerous, or non-neoplastic. So you really have to biopsy or understand that polyp. And probably if a polyp is not growing, if it is non-neoplastic, we would very, very much leave it alone. unless it is causing symptoms. If it was neoplastic, it needs to come out.
It is said nervous cells do not exhibit division as they undergo quiescence of cell cycle. And what if neurons around the gastrointestinal tract are damaged? Will they be able to grow back?
So that's an interesting question. I'm not sure I have the answer to that, but there are some experiments which people have done. Peristalsis always goes away from the esophagus, stomach, down towards the colon. So sometimes in the past, what surgeons have done is to take a loop of bowel and cut it. And if somebody has a very rapid intestinal transit, they cut it and reverse it. So you have an antiperistaltic loop, which then slows down transit when the intestine, because it's actively against the peristalsis moving towards the rectum. So I don't know if that answers the question, but that's the phenomenon that we know about.
I'm going to ask just another two more questions. One specific to gastroenterology and another one to the future of medical science. The first is from Balchandra Deshpande. Why in GERD reflux of food happens, and why does the nervous system detect this?
Right. So GERD is gastroesophageal reflux, where the stomach contents go back into the esophagus. Now, I very briefly mentioned that there are two sphincters at either end of the stomach. One incoming between the esophagus and the stomach, and the other outgoing from the stomach into the duodenum. The incoming one, which is the lower esophageal sphincter, really should function like a valve. It actually functions in response to this propagated signals which come down. And when food reaches the lower end of the esophagus, it opens, allows food to go to the stomach, but then it closes and prevents the acid and food from going back into the esophagus. So when that sphincter does not work properly, then you get reflux. I mean, that's a simple answer. It's much, much more complicated than that in terms of electrophysiology and manometry.
So I think the last question, which I think is an interesting question on how artificial intelligence and machine learning are going to change the field of medical science.
So the question is, how will it change?
Yeah, how will it change?
Yeah. So I think everybody is really, artificial intelligence is the buzzword nowadays. I mean, for scientists that want to get funding and move on in science, their specialized fields, artificial intelligence is the thing in medicine in particular. So essentially what, for instance, I have a colleague of mine whom I've given thousands of endoscopic images. And essentially they feed these images into a computer and say, this is this, this is this, this is this. The machine expects to learn from that. And then it gives an interpretation based on what we have fed it. But currently, artificial intelligence or this machine learning, the way it is what you have fed it, it will feed out. So it is, what people used to say, GI, GO. So if you put in something, that's what you expect to come back out. And right now, artificial intelligence is a long way to go before it matures, but it certainly could do a lot of things. We already use artificial intelligence when we use cameras which look at the small intestine. So these cameras, you swallow it in a pill, it goes all the way down the intestine, and it takes about 8 to 10 hours worth of recordings. we look at it to see if we can find ulcers or bleeding spots or something in the intestine. Now imagine running it at normal speeds and sitting through 8 or 10 hours to try and decipher what's inside that person's intestine. So we don't do that. We cheat a little bit. There's artificial intelligence which tells us, you know, you go to the spot, there looks to be something abnormal there. So those are current applications which are available.
Fantastic. So I'll end the Q&A here, if that's okay. There are a few more questions that are on here, and I'll send them to Dr. Ramakrishna, and he'll reply later. So before we wrap up, Chandrakant has something. Do you?
Yes. So this is just wanted to, thank you, Raj and Dr. Rama, so much for this. I just wanted to share the poster of the next event, and the title is Exploring the Brain, Its Birth and Function. And I think for, everyone who's heard today's talk, I think they are well prepped for the next step of the journey, as well as anyone else who will register as well. So yeah, you can register there. There's the link. We've also shared it in the chat windows and also in the YouTube comments. The title is Exploring the Brain, Its Birth and Function. It will be on Friday, the coming Friday at 5 p.m. again on the 11th of September. And we have the speaker is Dr. Bhavna Muralidharan, who is an amazing scientist and has made a career out of neurobiology. So please do not miss what she has to say. And of course, Dr. Raj Ladhar will be back with us then as well. He'll be here throughout the series to take the questions and talk about the speakers as well. So thank you so much. And I'm going to just hand it back to Dr. Raj to just wrap up the session.
Yeah, so thank you, Chandrakant. And very, very warm and hearty thanks to Dr. Ramakrishna for leading us off. It was a fantastic talk about the history of medical science and how the knowledge of how the different organs interact and talk to each other, how vessels and neurons and connect, and how the knowledge that we got to understand them was gained. I'm sure all the audience and all the participants are clapping for Dr. Ramakrishna. Thank you very much for spending this hour and a half with us. So everyone else, participants, I'm looking forward to seeing you next week when Bhavna will talk about the brain. So thank you very much.
Thank you. Thank you so much.
Thank you.
Thank you. So I'm ending the call now. The video will be there on YouTube for anyone to see till YouTube is there in the universe. So thank you so much. Bye-bye.
Bye. Thank you.